So next person doesn't really need that much introduction. I would imagine that most people in the room already know Anja, but yeah, Anja's going to give us an update on the mouse model.
Okay, good morning everybody. I'm very pleased to tell you a little bit about the work we've been doing as part of a large long COVID horizon research project, sort of me dragging or taking out some of the data that has been collated in a collaboration with James Stewart's group and also Andrew Owen from Liverpool as a pharmacologist and my group of veterinary pathologists mainly in Zurich. So it's about antiviral drugs that are actually used or have been used in clinics and their potential effect on viral burden and dissemination using a very defined, and you all know it, mouse model, the humanized ACE2 model. Is something happening here? No? Oh, yes, I wanted to go backwards, sorry. Now, we know there are a lot of neurological, neuropsychiatric symptoms in acute COVID-19 and also in long COVID. But, of course, it's also a lack of opportunity. I can say that as a veterinary pathologist. But what we know is brain infection is rare or has rarely been demonstrated in human patients because it relies on fatal cases, let it be of, and usually acute COVID. But the mouse model we use that really has a quite consistent widespread neural infection following intranasal inoculation with the VOCs. And it can be associated with a mild non-suppurative meningoencephalitis, as you can see it here. And I highlighted it, it's like symmetrical and it stretches everywhere. And only the cerebellum is usually spared. It takes quite a long time until you find the viral antigen there. So our question was whether WHO recommended antiviral drugs have an impact on viral dissemination to the brain in our mouse model. And we used delta and pangolin HB at different doses in this model and then had a look mainly at in situ level, but then also at qPCR level when a range of antivirals have been used. I come to that now. So we have nirmatrelvir, for which there is a strong recommendation in favor of the use in non-severe COVID-19 for patients at high or medium risk of hospitalization. Now we have remdesivir and molnupiravir. For these there has been a weak recommendation. Most of these nucleoside analogues, I'm not going to go into detail, you know much better, or the pharmacologists, of course. But we also included apilimod, because apilimod, I think, is interesting, and so far there is no current recommendation in favor or against its use, but looking how it works, it will not affect viral replication, airway, and lung, but should prevent productive neural infection of it due to its mode of action. So let's start with apilimod. What we have here is that I went back to material from one study where the drug was dosed 1 hour before infection and then up to day 4, and the animals were culled on day 5. So as you can see here, there was really no effect at all. So we found on day 5, we found the virus in the form of viral antigen quite widespread in the brain of these mice, and there was no effect as expected on the viral loads in nasal turbinates and lungs. So when we looked at remdesivir, we had a similar approach just a day earlier. Day 4, I have to say, is when you don't see viral antigen consistently yet. It takes time for the virus to get from the nose into the brain of the mice. So we saw an effect on the viral load in the nose. That has dropped, but not in the lungs at this time point, day 4, 1 day after treatment. But the majority of mice still had virus in the brain. As you can see, following on, it's usually, I mean, at least what we see, it seems to be almost solely via the olfactory epithelium and then towards the olfactory bulb, which is where we found the virus in the first instance in this group of animals. So then molnupiravir, this is a bit sort of, I found a bit more interesting. So we treated from 24 hours before intranasal challenge to day 4, but then the animals were culled only on day 7. And we saw an effect on viral loads in both nasal turbinates and lungs. And in none of these animals could we detect virus in the brain. Unfortunately, there was no PCR done, so I cannot claim that the virus didn't get there. But for me, it's an indication on how how actually these drugs can potentially block or affect the virus spread. I'll come back to that in a minute. So nirmatrelvir, we developed a bit more of an interest in, and we have by now done 3. Oh, I changed to lecture mode, so you will understand. No, it works again. I show you this now. So the first one was that the animals were treated 2 hours prior to challenge, and then up to day 3 and culled on day 4. You remember that's an early time point to detect virus in the brain. We had evidence of a reduced viral load in the nasal turbinate, but virus spread in a third of these animals. So whether that's due to blockage of spread in general, or whether the virus was just not there yet in a sufficient amount to be detected by immunohistology, we cannot say. So the next experiment was that we had a look at treatment 1 day prior up to day 3 after infection, and then we let the animals sit up to day 7. So you can see that there is a reduced viral load in the lung. The nasal turbinates weren't examined in this case. We found viral antigen in the brain in 3 of these animals, but there was sufficient virus in the brain in all of those. So it didn't block infection. But what we could see was that the average loads in the brains was higher than in the lungs. And you can see that on the left as well. But when the virus was seen in the brain as viral antigen, the distribution was just the same. So we had a look at the RNA-seq data we generated from that. And you can really say that these brains fall in with the virus-positive and negative brains. So there was really nothing spectacular about this. Of course, we have the upregulation of the inflammatory markers in virus-positive brains. And overall, the inflammation seemed to be a bit at a lower level than in the non-treated animals. But my interpretation would be that we just delayed brain infection, because Andrew then said, this is a drug that has an extremely short half-life. So you can expect it to be eliminated within maximally 24 hours. So of course, us only treating up to day 3, there was plenty of time. There was another 4 days for the virus to pick up speed in the nasal cavity and then go into the brain. So the next approach, which I think is the best one and the one we want to pursue further, is that we actually treated all the way through, and the animals were culled 30 minutes after the last dosing to be sure we have sufficient drug in there. And there we could see a reduced viral load in the brain. At day 7, there was no effect, no significant effect on the lungs, but really here now in comparison, the average viral loads in the brains were lower than the lungs. Still, there was detectable viral RNA in the lungs, yeah? And in one of these animals, we also saw virus antigen, and that did not look different. But overall, there is an effect that we see, but as I said, it's treatment up to 30 minutes before culling. So how could the treatment work? I think it's a clear story that you can see here. So I'd say there's a reduction of viral replication in the olfactory epithelium. You know, mice have lots of olfactory epithelium rather than the normal nasal mucosa because they have to smell very effectively. They have to pick up anything. So it's simply less opportunity for neurons to become infected because there is neurons directly from the olfactory epithelium, and then they go into the olfactory bulb. And you can see this here. So we would have here a few infected epithelial cells. It goes through. We find here some axons infected, and then it moves sort of on, and eventually it's widespread in the brain. And in the brain, it's neuron to neuron spread. There has been work, at least in vitro, that has shown that. So I would say there is no evidence that any of these drugs reach the brain parenchyma. And I think it's in particular the apilimod results that show that, because apilimod should prevent productive neural infection if it got into the brain parenchyma. So, I mean, if they won't pass any blood-brain barrier if they're if they are in the blood. So we will have a closer look at nirmatrelvir. We're currently in the middle of a pharmacokinetic study and see whether we can detect it in the tissue. But of course, you can imagine it, if any drug is in the blood, we will detect it in a tissue sample. It doesn't mean that it's in the parenchyma. It can still be in the blood and never get into the brain parenchyma in this sense. So that's just to acknowledge all the collaborators and the groups working here. So the two small figures are Janine and Simon, the doctoral students in Zurich involved. And for any of you who are interested in the long COVID project, that's funded by the EU, and because Switzerland cannot be part of it at the moment, by the State Secretary for Education, Research and Innovation, so that pays my part. It's a big study on patients, very focused on that, and I'm running the small part of the mechanistic studies in the in vivo model, and that's how I dragged James' group into it. So thanks for your attention.
Thanks, Anja. Right, any questions? Yep, straight in there.
Thank you, really enjoyed your talk. I was just wondering, with your mice that have all this virus infection in the brain, do you see any neurological clinical signs of infection, like ataxia or walking in circles, anything like that?
Not with the doses that we have. They just lose weight and then you reach clinical endpoint. Cool. So it's probably before that. Also, it's a neurotropic virus, but I wouldn't call it a neuropathic virus. When you look at this, and we are currently finalizing even ultrastructural studies, because you don't see anything at light microscopical level at the doses that we work on, the neurons look perfectly fine. It will have a functional effect, but it doesn't kill the neurons. Yeah, so I call it neurotropic, but not neuropathic.
Just very quick follow-up. Do you see much inflammation of the brain?
That's what I showed in the beginning. You see none or a mild, non-suppurative, the occasional neutrophils, like with every virus infection, encephalitis. It's very mild, and it's directly associated with the presence of a lot of infected neurons. Yeah.
Lovely. Thank you.
Yeah, thanks very much. Very nice talk. Have you looked at the sequence coming out of the brain? Is the virus still infectious? Can you uncover infectious virus? I worked on measles, SSP for many years, and obviously the mutations that happen in neurons, then, you know, you don't always can't get infectious virus.
We have the material, and plans are to actually compare the virus in the lung and in the brain. Well, we haven't done that yet, but that's part of what will come. The project is not finished yet.
Any more? No, I think we're good. Right, okay. Thank you again.